My name is Jonathan Chou. I'm a third-year medical oncology fellow in the Feng and Ashworth labs at the University of California in San Francisco. I take care of patients with advanced prostate cancer and conduct basic and translational research in the laboratory to help improve our understanding of this disease. Many of us probably know a family member or friend who's been diagnosed with prostate cancer. And while the majority of patients who are diagnosed with localized disease can be cured by either radiation or surgery, patients who develop metastatic disease, in other words, disease that's spread to other parts of the body, have an average life expectancy of just under 4 years. Perhaps many of us will also be surprised to learn that nearly 30,000 men in the US this year will die from advanced prostate cancer. I've taken care of men from their early 40s to their late 90s with advanced prostate cancer. And the treatments that I can typically offer my patients are those that have been around for a long time. Androgen deprivation therapy, also known as castration, radiation particles, and cocktails of toxic chemotherapy drugs, many developed decades ago. And while there have been several newly FDA-approved therapies for the treatment of metastatic prostate cancer over the last few years, each of these therapies only extends survival by about a few months. And they're also given in a sequence not optimized for each individual patient. While the general consensus guidelines that have been developed provide a great starting framework for how we should think about treating prostate cancer, they fail to take into account the individual and unique mutations within each patient's tumor. As we are beginning to learn more about the genomic and molecular alterations within prostate cancer, we are learning that prostate cancer is really a group of multiple molecularly heterogeneous subsets of diseases. Our lab recently sequenced the whole genomes of over 100 metastatic prostate cancer biopsy samples taken from men who had progressed on standard of care therapies. And what we found was a spectacular array of genomic structural variations and genetic changes that underlie each individual patient's tumor. So I ask you, why then should one type of therapy or one sequence of therapy fit all patients when we know that the building blocks for each patient's tumor, the molecular changes that are occurring, are so unique amongst our patients? Fortunately, as we're discovering and learning more about these molecular alterations, we are discovering new ways of targeting these specific mutations. One class of mutations that frequently occur are in a family of genes that help normally repair DNA damage, from errors that occur during DNA replication or from damage, double-stranded breaks, that occur from environmental exposures. Now, tumor cells can benefit from losing these normal DNA repair mechanisms because it gives them an increased ability to mutate and to adapt to our therapies. So, for example, about 15% of patients with advanced prostate cancer have a mutation in a gene that some of you may have heard about called BRCA2. Now, while losing BRCA increases the predisposition for developing cancer, it also exposes a key therapeutic vulnerability. BRCA2 mutated cells are exquisitely sensitive to a new class of agents called PARP inhibitors. It's the Achilles heel for these BRCA2 mutated cells. And so while these tumors can grow, we have a way of targeting them that's more efficacious and less toxic than traditional chemotherapy. And this class of agents are actually currently in clinical trials for men with advanced prostate cancer. Similarly, another DNA pathway that's commonly mutated is in a class of enzymes called mismatch repair proteins. Now, while losing these particular proteins leads to the generation of multiple abnormal proteins in the cell, it also exposes another Achilles heel. These proteins can actually function as neoantigens to stimulate the body's own immune system to search, destroy, and target these cancer cells. In fact, the FDA approved its first tissue-agnostic therapy called checkpoint inhibitors, a type of immune therapy for any tumor type that had lost the ability to undergo mismatch repair. So the underlying biology of the cancer, whether it was a prostate cancer, a lung cancer, or a colon cancer, revealed that there's a less toxic and more efficacious treatment for our patients. In my own research, I've been focusing on a DNA repair gene called CDK12. And what we've shown is that loss of CDK12 leads to widespread genomic instability. And this seems to lead to sensitivity to both PARP inhibitors and checkpoint immune therapies, which we're currently evaluating in the context of a clinical trial that we're running at UCSF. Now, why does this matter? Well, it turns out that about 8% of men with advanced prostate cancers will have a mutation in CDK12. Another 5% of women with ovarian cancers also carry this exact same mutation. We are beginning to learn that not only do these CDK12 mutated tumors not respond to our traditional types of therapy, but they actually might respond better to these targeted approaches and to immunotherapies. We are beginning to dive deeper into the biology and function of CDK12. How does it regulate the DNA damage response? How does it regulate DNA replication during cell cycle? And how does it regulate transcription, splicing, and gene expression in these cells, with the hopes of trying to understand new ways of potentially targeting this molecular subset of cancers. In addition, using CRISPR-generated CDK12 knockout cells, we are conducting genetic and chemical screens to try to find new ways and new drugs that could be potentially used. As we enter an era of medicine where we can profile tumor genomes, transcriptomes, and proteomes at single-cell resolution, we are beginning to learn that there is a tremendous amount of information at our disposal that we can potentially use to make better treatment decisions for our patients. The goals and the vision for precision oncology over the next 10 years are to be able to match patients with specific sets of mutations with the right kind of therapy that's less toxic and more efficacious. And it's starting to become a reality. The FDA approved a new class of drugs called NTRK inhibitors last year for the treatment of any tumor type that carried an NTRK gene fusion. So instead of thinking about tumors based on their tissue of origin, by understanding the molecular alterations, we can start to develop better and more targeted therapies. It's one of the exciting things about being an oncologist. And I hope that in the next several years, as we start learning more about these molecular alterations, we'll develop new synthetic lethal approaches to improve the lives of our patients living with metastatic cancer.